Hello, and welcome to lecture on 3D structure prediction of proteins. Your reading is Xiong, chapter 15. So today we're going to be talking about homology modeling, threading, ab initio, or namely molecular dynamics methods, mutual information and direct contact analysis. And then later, Arne Elefsson is going to talk to you about deep learning. Okay, so what's homology modeling? Homology modeling is something that we can do if we have a target sequence, but no structure, but we can find a reasonably close homologue for which a structure is known, right? So basically all we know about our target is, let's say, the sequence. I'm just making this up, some sequence. So we do a database search of some suitable structural database, probably the Protein Data Bank. And this could return 0 hits. It can return 1 hit. It could return more than 1 hit, right? Our next task is to select a template. Actually, modern methods can and will use multiple templates. In fact, I have a paper where we use multiple templates to solve RNA structure. But for purposes of this discussion, we're going to just select one template. And so, let's say we apply the criterion of lowest RMSD, or maybe we wanted a template that was most complete in terms of number of residues that are resolved. So whatever we choose, we whatever criterion is, we choose one of these. And let's say our template looks like this. So we're gonna align our sequence to the sequence of this template, and that's going to give us some sort of initial alignment. That's alignment again based only on sequence, but we do have structure, right? And so there's certain things that we probably know about this structure. Like, for example, let's say it's a structure that has a known ion binding site. So let's say that there's some sort of ion binding triad, maybe histidine, and a glutamine. And maybe we know the position of this same motif in our target sequence, right? So if within our alignment we see that this important motif is not lined up, well, that's a good reason to then go and refine our alignment, right? And actually, anytime you have a pairwise alignment, you should check it manually. But in this case, let's say we checked it and we found that we can insert some gaps here and there and get a better alignment. So we do that. Okay. And then after that, our next job is to build a core. And building the core basically means copying the backbone coordinates directly from the template and basically renaming them and renumbering them. So now they're the sequence and with the correct numbering of the target. And this would look something like, let's say, like this. Here's the beta sheet, and then here's the alpha helix, and you notice there's no solvent exposed loops, and there are no side chains, right? So just the backbone and just in the core. Now, why do we build the core first? Well, there are a couple of reasons for that. Number one, the core is more packed and less mobile, and so our nuclear coordinates are known more accurately, because anything that's mobile will be averaged out in the experiment most likely, and on the other hand, if it's closely packed, then the thermal oscillations will be much smaller, and we'll be able to observe them with greater precision. And then also the core is usually more conserved in evolution, so our alignment will be superior, and it's harder to build a core. The loops are easier just because they have more room available, and also the loops are less. There's less of a concept of a structure of a loop because they are mobile. Any number of solutions is as good as any other. Okay, so once you build the core, then your next step is to then go and add loops and sidechains. And actually, these two steps, the loops, the side chains, they're done with completely different methods. But anyway, like we say, we started out with this backbone only structure of the core, and we added these loops connecting these secondary structure elements, and then we started to build in our side chains. And there are unique challenges to both of these processes, and we'll talk about how we solve them. Then, one of the main challenges is that these side chains they mutate, right? So they're not the same necessarily, or certainly not in every case, as the residue types in the template. And some of these might be increasing in volume, for instance, and then we have to, if this increase in volume happens in the core, then we have to reaccommodate that residue and neighboring residues to make room for it. And there may be steric clashes, there may be even interlocking rings and things like that. And so we need to resolve all those issues. And we do that with typically very fast methods, which are not physically perfect. So then we need an optimization step. And optimization basically does things like take side chains which are maybe not interlocking, but the atoms are a little bit too close for comfort, right? And we're talking about problems on the length scales of tenths of angstroms maybe. And so optimization is basically dropping this protein into a molecular dynamics potential, which we'll discuss later, and letting the physics, turn on the physics, and then finding new positions for all these nuclear coordinates that are physically reasonable. And that means a local energy minimization. So you're only going to basically follow a gradient to a nearby minimum. We're not going to try to find a global minimum, which would involve maybe rearranging all the atoms in our structure. So it just gets rid of remaining small steric clashes, and at that point you would have a structure which can be dropped directly into a molecular dynamics package, and you can calculate you can solve the equations of motion and look at the motion of this thing without it blowing up. I mean, the reason I mention this is because if you don't have properly minimized structure, then the atoms being close together will generate large forces, right? Because there's the repulsive part of the Leonard-Jones potential, and this will mean that after one time step, these forces cause large accelerations, and the atom will fly apart. So once it's optimized, then you go to this last step, which is to evaluate, and you do things like look at the phi and psi angles and make sure that they're all in allowed regions of the Ramachandran plot. You look at things like bond lengths and angles and everything else that you would do even as an experimentalist to check the quality of your protein. Okay, so now let's get into this in detail. So, but before we go further into it, you appreciate at this point that homology modeling is something that you do if you have sequence and you're able to find a homologue of known structure, right? If you can't find a homologue of known structure, there might still be a fold which is the correct fold for the sequence, but which is not related by evolution, right? And for that, we need a fold identification method or threading. Then, if you don't have either of those, then you could try ab initio folding, basically dropping it into a molecular dynamics package and letting integrating equations of motion until it converges. The problem is that this is extremely expensive and only works for very small proteins. But these days, there is a new option that's coming to the fore, which is deep learning. And these methods have gotten so good that typically that I think you're going to start using these as a first resort. Even before you try homology modeling, even if you have a close homologue. And these deep learning methods take into account things like coevolution. They know about all the structures that have been solved, and so these like I say are going to probably become our first resort and last resort. They're just that good. Okay. So what do we mean by template selection? Well, the selection of a template is all about, well, number one, the quality of the template, how big it is, what sort of resolution you get, but also you have to worry about whether it's the right template, right? And so very early on, back in the 70s, Mike Levitt observed that there's a sense in which a structure is more conserved than sequence. And so what that means is that there's some point, depending on the author, this moves a little bit, but it could be maybe 30% or maybe 20%, somewhere around there, at which you start to lose the relationship between structure and sequence, or rather, where the sequence identity no longer implies structural similarity. And it's very controversial to say that I think, in my opinion, that structure is more conserved than sequence, because these are very different quantities. One is bounded, one is not bounded, for starters. But what they're trying to say is that if you have high sequence identity, then you can reasonably, very confidently assign structure. You can say, oh yeah, I can assign the structure, and looks like if I know the right answer, I determine that the root mean square deviation is less than half an angstrom, right? And that actually holds up pretty well. I mean, you can see here that if you're at 40%, then you're still very likely to have structural similarity of less than 1 angstrom RMSD, root mean square deviation. But after some point, 30, 20%, something like that, then your root mean square deviation starts to diverge, and at that point, the odds are actually against you that you found the right template, right? I mean, the right template may be in there somewhere, but there are thousands of possibilities, and you don't know which one is the right one. So we refer to this zone as a twilight zone, the zone where you just don't know that much. And in general, you're going to want to stay inside the safe zone. Okay, the zone in which sequence similarity basically implies structural similarity. Okay, we talked about realignment, and again, this is a process whereby you know your template structure, you know certain things about the biology of this protein. You know the start endpoints of your secondary structure. You know where certain, as I mentioned, maybe motifs, maybe catalytic sites lie, and you want to make sure that anything that you know about your target sequence is consistent with this structure, right? So you might need to shift your alignment a bit so that maybe this ion binding site lines up with the one in the template. Then you build a core, and as I mentioned, that's basically copying the backbone coordinates of the core and throwing away the side chains, right? And so then you're going to have to rename these residues and renumber them, and then basically you have the core of your target, hopefully. So that step is actually pretty easy. But then it gets a little bit harder when you actually have to start building in your side chains, because some of these side chains are going to be on the surface, and so there's going to be plenty of space for them to move around. Maybe it won't be so much of a problem, but deep in the core, you have a very crowded environment, and you can have things like residues where the atoms are too close together, and you can even have one residue side chain going through the middle of a ring, or doing something really crazy like that. And so there are methods to not only build the side chains, but then to alleviate all of these steric clashes and other chemical issues. And mostly they're based on a rotamer search. So rotamers are stable, like integer numbered, stable values of the dihedral angle, right? So this is your dihedral angle. Let's call it phi. In this case, this C2H6, these hydrogen atoms like to sit in the middle position with respect to the hydrogen atoms on the other end, and so so in this case, you would have minimum energy positions at 3 angles between 0 and 2 pi, right? because the hydrogen atoms on one side, like I say, want to sit in the middle between the hydrogen atoms on the other side, so they have one minimum energy position at, let's say, phi is equal to 0, another one at 120 degrees, and another one at 240 degrees, right? So we call those rotamers. In this case, they're basically 3 rotameric states, and they're integer numbered, right? And they're different, these rotameric states, and the number of rotameric states, that their phase offset, are different in principle for every bond in your protein. But of course, you can just make a determination based on the atom types and how they're connected, what sort of rotameric states exist, and then you explore these. And since they are integer numbered, it's really not all that expensive to do. And the way you do is you start with the residues that are actually engaged in some sort of steric clash, and you rotate them until you find some position that's more comfortable. But then sometimes you also have to rotate the side chains of neighboring residues. And it sounds a bit complex, but actually it's usually done very quickly. So one popular program for doing this is called Squirrel. And but the same idea has been implemented in many methods. For example, the stability prediction potential Foldex does this for you. So that's a solved problem. And then you have to go and solve the problem of your surface loops. And there are a couple of ways to do this. So one way to do it is by fragment assembly. And the way the fragment assembly works is you identify the loop that you want to place, and you identify anchor points. So these are bonds that are resolved in your protein, and you need the distance and the relative angle of these two bonds, like this. And then you need your sequence that you're trying to fit in here, whatever that is. And you have a database of structures, and you're going to search that database for structures that have roughly similar sequence, the same length, and these anchor points that match up with the ones in your protein, right? And that might return many hits, right? Many more or less suitable loops, and you might try all of these and pick out one that's the best match, right? That matches well with the anchor points, that has a similar sequence, and very importantly, it doesn't have very much in the way of steric clashes with neighboring loops or with the core, right? And so you chose one of these and you stick it in, and that's it. And this is a reasonably fast method, and it gets reasonably good results because it's a these are actual experimentally observed loops. And another approach would be the analytic approach. So the Lotome lab was actually next door to the Altman lab where I worked as a postdoc. And so I've talked to these guys, and they what they can do is they use motion planning, which is a field of robotics, to find an optimal path connecting these two anchor points. And it turns out that these two anchor points are separated by 6 degrees of freedom, right? Because there are x, y, and z Cartesian degrees of freedom. And then there's also rotation about x, y, and z. And so that means there's 6 degrees of freedom separating the anchor points. And that means that a loop that is long enough and that has 6 degrees of freedom can be found which connects these two anchor points, right? And so each residue in a protein has 2 degrees of freedom, right? The phi and the psi angles. And so if you have 3 residues in your loop, then you're guaranteed to find a combination of phi and psi angles that connect these two anchor points, as long as the anchor points are not too far apart or pointing somewhere weird away, right? So 6 degrees of freedom. And you can actually use similar methods even if you have more than 3 residues. But 3 residues is the minimum to guarantee convergence. And this method is also very fast. And lastly, you could do ab initio, meaning that you could just sort of somehow or other patch in a sequence and then let molecular dynamics minimize the positions. But actually, this is probably has the worst convergence because if there are any steric clashes, it might just explode, and it's more costly than all of these other methods. So it's not recommended. Most of the time, you'll use either fragment assembly or an analytic method. Okay, and then you have to do minimization, right? So at this point, we have not only the surface loops, but we also have built-in side chains, and we've even resolved some of the steric issues. Although there's still going to be problems with atoms that are maybe not severe steric clashes, but maybe a little bit too close for comfort, right? And these are distances that are wrong on length scales of maybe tenths of angstroms, right? But which is enough again to make your molecular dynamics program crash if you were to drop this in. And plus, it's just unphysical, so you want to report a good result. And so what you do is you drop this into a molecular dynamics package and do a local minimization. And that is like I said, we're going to get into molecular dynamics a little bit later, but basically we're going to solve, we're going to follow this something like a steepest descent of this potential to find a local minimum. In other words, a nearby minimum. We're not looking for the global minimum. We're just looking for something that's more comfortable than where we are now. And this could be the most computationally expensive part of this process, but if the sidechain building and the loop building went okay, then it should probably converge. And then lastly, we want to evaluate the model. We want to do things like check that the phi and psi angles are in allowed regions of the Ramachandran plot. We want to make sure that the bond lengths are all correct, close enough to the minimum, and that there are no steric clashes. So, so like let's say one of our residues looks like this. There's a nitrogen, there's a C alpha, there's a carboxyl, and then our next residue, and this has some sort of side chain atoms, and and this has some side chain. What we're going to check is again phi and psi, and we're going to look at the lengths of these bonds and make sure that they're reasonably close to their equilibrium length, and we're going to take another look again at make sure that these interatomic distances are not too close. And there are standard packages that do this. And they're basically the same packages that maybe a crystallographer would use before submitting their experimental coordinates to the PDB. Okay. So that's all I'm going to say in this part of the lecture. And now you would go to part 2 of the lecture. I'll see you there.